Alright guys, let's talk about enzymes. Specifically, energy and enzymes. So what we are going to talk about here are, well, we're going to talk about energy first, so our different types of energy, including kinetic and potential energy. We'll define those. Then we will get into our types of enzymatic reactions and how those enzymatic reactions use the different types of energy. That will be part 1 of this video. Part 2 will cover catabolic and anabolic reactions, along specifically with how enzymes work and how we can prevent enzymes from working, enzyme inhibitors. So let's jump right in. Let's get into the different types of energy. All right. So our two major classifications of energy in life, in physics, in the universe, include kinetic energy and potential energy. And if you remember way back from the beginning of the quarter, the definition of energy is the ability to do work. The ability to do work. And what we can really ultimately translate this into is the ability to move things. All right. So kinetic energy is that movement. So kinetic means movement, right? Like kinesiology is a study of the different movements that the human body can make, like joints and muscles and things like that. So kinetic energy is the energy of movement of a system. And when we say a system, we can refer to a molecule, we can be referring to two molecules, we can be referring to a cell, we can be referring to a whole organism, a whole planet, the whole universe, right? We can define what a system is. One form of kinetic energy is actually heat. Heat is the energy of molecules vibrating. And the higher the heat of something, the hotter something is, the more those molecules are vibrating. The lower the heat, the less the molecules are vibrating. So when you touch something really, really hot, what you're feeling is just extreme vibration. Something that is at absolute zero, we say 0 degrees Kelvin. So something that's at absolute zero, the coldest anything can go, has no motion at all. Radiation is another type of kinetic energy. This is the energy of photons moving through space. And photons are particles of light. They look like, we like to depict them as waves, right? Photons are waves. They come from the sun, they come from our lamp, right? They come from the headlights of our car. They are moving through space, so they are a form of kinetic energy. And radiation energy includes not only radio waves, right, that you listen to in your car, it also includes visible light, the stuff that we use to see. It also includes microwaves and X-rays and gamma rays and infrared and all those. Those are all different forms of radiation. And some radiation is harmful, some radiation doesn't hurt us, doesn't affect us at all. We'll get more into this when we talk about photosynthesis and the different forms of radiation. Because as it turns out, photosynthesis depends a lot on radiation and kinetic energy to work. Potential energy, on the other hand, potential energy is more stored energy. It's energy that you're not doing right then, that you're not using right then, but you have the capability to use it, and you have the capability to convert it into kinetic energy and movement. So, one example of potential energy is chemical potential energy in particular. This is the energy stored in a chemical bond, and that chemical bond can be between C double bond O, as I've drawn here. It can be C to C, it can be C to OH, right? Oop, my OH got a little bit screwed up there. There we go. All right, it can be O to H. Any chemical bond. All right. Now we're gonna get into this in the later part of this lecture. The more oxidized the bond, the less energy in that bond. We'll get into what oxidation is later. But just tuck this away in the back of your head for right now. Another form of potential energy is electrochemical potential energy. This is the energy in an electrochemical gradient. Remember, an electrochemical gradient is an uneven distribution of ions across a barrier. So here we have way more hydrogen ions on one side of the barrier, way fewer on the other, there is an electrochemical gradient. There is energy there. When that hydrogen is able to flow across the barrier, say we put like a transport protein in there, you actually do release energy as hydrogen flows down that electrochemical gradient. You release that potential energy and you convert it into another type of energy. And there's many more types of potential energy than what I have here. There's more types of kinetic energy than what I have here. These are just the ones that we are going to deal with most often in biology. So we kind of implied this earlier, but energy can be transformed from one state to another. So you can go from chemical potential energy, or just plain chemical energy, so chemical potential, or just plain chemical energy, to heat, so to kinetic energy. You can convert one energy type to another. But we have rules that we have to follow when we do these conversions. And these rules are called the laws of thermodynamics. Thermo, if I can get this to work properly, thermo means heat, dynamics means movement. Heat is kind of a universal form of energy. It's a very, as we'll see later, it's a very disorganized form of energy. So we kind of use it, the word thermo as a catch-all for just describing energy. So thermodynamics are the laws of thermodynamics are the rules that govern how energy in general, not just heat, behaves. And there's two main laws. Some physics instructors will even say there's a third law. But we are just going to look at two main laws here. Third law. Okay, dang it, keep doing this, don't I? So the first law says that energy is conserved. Whenever you transform energy, you can't make more out of thin air and you can't just get rid of it. It can't be created or destroyed. It has to be conserved from one type of energy to another type of energy. I mean, this kind of intuitively makes sense. You can't just lose it, right? The second law says that entropy increases as a net, so that as a whole, when you take into account all the different types of energy transformations that are happening. So what's entropy? Entropy is disorder. Or you can also think of it as chaos or useless energy. Energy that you can't harness, you can't utilize to do work. And most of the time, entropy comes in the form of heat. Heat is very disorganized. We can't really use it to do things. We can't use the energy and heat to move things very, very easily. So most of the time when we say entropy increases, we mean that heat is being released, heat is increasing. So we are transforming many of our different energy types into heat. So what this means, as an example, if we take some nice, relatively high quality chemical potential energy, say we take a COH bond, and we transform it into kinetic energy, say in radiation, right, in a photon. So say we burn that COH bond and fire releases light. So we're releasing some of that COH bond energy, that chemical potential energy, as light, as radiation energy, kinetic energy. Well, a good chunk of that energy that you release won't actually be released as the light that we want. It'll be released as this useless heat energy. So we are increasing entropy. We are increasing the useless amount of energy in the universe. So this doesn't happen for absolutely every single chemical reaction that you release heat, but when you sum all the chemical reactions that are happening together in the universe, then yes, as a whole, heat is being released, and you are gradually degrading your high quality energy sources like COH into lower and lower quality energy sources like heat. Entropy is increasing over time. And actually, give me just a sec. Oop. What am I doing here? There we go. I'll be right back. Go. Okay, picking up. Sorry about that. We were talking about the laws of thermodynamics. Now, there is a third law of thermodynamics, and that third law says that anything that is at absolute zero, that's 0 degrees Kelvin, has 0 entropy. The whole point of the third law is just to establish a baseline to say, here's where there is no entropy, when you're not moving at all. Alright, so don't really worry about the third law too much in this class. So we can transform energy, as we just said before. Here's a couple really good examples of transformation of energy. Alright, let's start with sunlight, specifically ancient sunlight. That is, of course, radiation. Sunlight's in the form of photons, and that's a form of kinetic energy. Using the power of sunlight, plants, through the process of photosynthesis, will take in carbon dioxide and water, plus the photons from the sun, and make this molecule right here. And this molecule is, of course, our good friend glucose. So we take in energy, we take in radiation energy, and we convert it into chemical or potential chemical or chemical potential energy, whichever order you want to put that in. Now, ancient ferns did this really, really well. They did photosynthesis really well. Built up lots and lots of chemical potential energy in the bonds of their glucose. Now put the remnants of those ancient ferns under lots of heat and pressure, and we would convert that glucose into this molecule right here, C6H12. That is actually octane, so that's oil and gasoline. Now there are a couple steps between glucose and oil and gasoline, but that's the idea. So we are converting chemical potential energy of one type into chemical potential energy, right? Just a slightly different variant of that chemical potential energy. And then we're going to take that chemical potential energy in the gas, and we're going to put it through a chemical reaction using a spark plug and convert it into CO2 and water. And that's what we do in our car. Now the chemical potential energy that we release from this gasoline goes mostly towards the kinetic energy of our car tires moving. But our engine also gets hot too. So entropy always has its due, right? Almost every time you convert energy, you will increase heat, or you'll produce heat as a result. So along with your car tires moving, your car engine will get hot as a result of converting the chemical potential energy in oil or gasoline to the kinetic energy of movement. Alright, so here's where we're going to come back and we're going to talk about the different types or the different qualities of chemical potential energy. So right here, I'm highlighting this C to H bond right there. This bond right here is very high quality, has lots and lots of chemical potential energy. In contrast, this bond over on the far right, this C double bond O bond over here, has very little chemical potential energy. As we move from C to H, down this, what I call an oxidation ladder. This is an oxidation ladder. As we move down from left to right, we are decreasing our chemical potential energy. So we are decreasing our chemical potential energy. Conversely, if we're moving from left to right, we are increasing. So that's what I have down here, increasing chemical potential energy as we move from did I say left to right? I meant right to left. Alright. And part of the reason is because you probably guessed it's the presence of this polar covalent bond right here. All right. So if we think about a polar covalent bond, let me go back. All right. Think about this bond right there. Okay. It's somewhat polar covalent, but not too much. The electrons in that bond are probably hanging around over there. Okay, closer to the carbon than the hydrogen, but not all that much closer. If we look at something like this, though, a C to O bond, well, those electrons, they're going to be hanging a lot more around the oxygen instead. Oxygen's more electronegative. This bond right here is easier to break. So it yields less energy for us because it's easier to break. So the harder a bond is to break, the more equally shared those electrons are, the higher energy in the bond. At least that's how we can think of it. Okay. So that also holds if you create a double bond with the oxygen, or if you're a carbon and you have a double bond with an oxygen and then a bond with a hydroxyl, yet another oxygen. If you have two double bonds, man, you're as low energy as they come. We call this movement from left to right, from having lots of energy to having not so much energy, we call that oxidation. So as we go from CH to COH, we oxidize. The opposite is called reduction. So as we're moving from right to left, as we're going from COH to H, we are reducing. So reduction to the left, oxidation to the right. We'll get into why it's called reduction and oxidation on the next couple slides. To use an example of this, look at this molecule down here. Anybody recognize this? This is actually a fatty acid. Specifically, it's a saturated fatty acid. This molecule right here, this is glucose. This molecule right here, you probably don't recognize this one. I'd be pretty impressed if you did. This one's called pyruvate. We're going to learn what that guy is in later this week. And of course, this one over here, this is carbon dioxide. So fatty acids have way more chemical potential energy, look at that, lots of those CH-CH bonds, than glucose. Right there, there's a COH bond. There's a CO bond right there, there's a COH bond. Right? Pyruvate, on the other hand, has even less chemical potential energy than glucose, C double bond O. Here's a C double bond O. That minus can be substituted with an H. So right there, that little minus, you can mentally add an H to it, dang it. You can mentally add an H to it, okay? So that's a C double bond O OH, like right up here. So that's very, very low chemical potential energy, and then of course we have CO2, as low as they go. So this explains something. You may know, maybe if you took nutrition, or maybe if you're in the habit of reading the nutrition facts on the back of a cereal box, fat has 9 calories per gram. So for every gram of fat that you eat, you get 9 calories from it. Sugar, like glucose, has 4 calories per gram. Pyruvate, if you were to eat it, has even less than 4 calories. I don't know how much. And carbon dioxide, of course, has 0 calories. You can't live on carbon dioxide alone. So fat has more calories simply because it has more higher energy carbon to hydrogen bonds. Alright. So we took a little detour there. We are going to circle back to that. Now we are starting to zero in on how all this relates to biology. Okay? So in order to get into this stuff, first let's define some things. Metabolism. We have seen this word before. Here's the definition now. Your metabolism is all of the chemical reactions in your body, all of them, or almost all of them, are done by enzymes. All right. And these chemical reactions aren't done all at once in one big fell swoop. They are done step by step. And you build up or break down these very, very complex molecules little bits at a time, step by step, through metabolic pathways. So metabolism is a group of chemical reactions, sometimes a couple dozen. In contrast, a single chemical reaction is just one step. Metabolism is several steps. Chemical reaction we can define as the rearrangement of atoms and electrons. Sometimes we release energy or absorb energy as well. So that relates back to our whole entropy and first law and second law. We can split metabolism into two halves. This is kind of like two halves of the same coin. All right. Catabolism is the breakdown of molecules over many steps. Anabolism is the buildup. So catabolism is breakdown, anabolism is buildup. So we say that in your catabolic pathways, you break down molecules. And this particular chemical reaction, hydrolysis, is often found in catabolic pathways. On the other hand, you can probably guess where I'm going with this. Dehydration, or condensation, reactions are one of the many types of reactions that can be found in anabolic pathways. With catabolism, because you are breaking down molecules, you're breaking the chemical bonds in molecules, usually you are releasing energy, and this, I'm saying producing energy here. Remember, energy can't be consumed nor created nor destroyed. So my wording here is a little bit off, but you get the idea. You're releasing energy with catabolic pathways. With anabolic pathways, you're building things. It takes energy to build things, right? So you're going to take in energy. And when I say energy, we can be talking about kinetic energy, potential energy, any type of energy here. Just depends on the particular reaction. And here's how I remember the difference between catabolism and anabolism. Cats, catabolism, destroy things. Love cats, but they are destructive little buggers. So catabolism is the breakdown or destruction of things. Anabolism, when I think of anabolism, I think of anabolic steroids. Anabolic steroids are the ones that give you lots of muscles. They cause you to build muscles. Anabolic steroids actually, so here's how they actually work. They make you do gene expression more, so you produce more proteins in your muscle cells. So your muscle cells get packed full of protein and they bulk up. They actually get packed full of cytoskeleton protein. So not only do they bulk up, they also get stronger. So with anabolic steroids, you're building up your muscles. So anabolism is the buildup of molecules. Okay, so here we are looking at an individual reaction now. And enzymatic reactions, a lot of times we can use this term interchangeably with simply a chemical reaction. RXN means reaction. In biology, it's very rare to see a chemical reaction that happens all by itself. We almost always need enzymes to help us do those chemical reactions. So a lot of times whenever you hear the word chemical reaction in biology, you can assume that there's an enzyme implied in there somewhere. So here's an example of a chemical or enzymatic reaction. Here we're taking sucrose. We're adding in water. The enzyme sucrase is going to add the hydroxyl from that water to one half of the sucrose molecule and the hydrogen to the other half. Here's what sucrase looks like right here. Remember, it's an enzyme, so it's a big protein. Here's sucrose right there. So this single reaction is one step in the full breakdown of sucrose. So as an example, you're going to take sucrose. In the first step, you're going to hydrolyze it into glucose and fructose. This is a hydrolysis reaction. But this is only the very first step in the breakdown of sucrose. We've only seen one chemical reaction. We're going to take that glucose and fructose and we're going to send it through another reaction called a phosphorylation reaction. And we're going to make a molecule called glucose 6-phosphate. I'm shortening it to just G6P for now. You do not have to know this, I'm just giving you an example. At least you don't have to know it yet. So this is a phosphorylation reaction. We're going to learn what phosphorylation reactions are in about 10 minutes. And we're going to take this G6P thing and we're going to convert it into fructose 6-phosphate through yet another type of chemical reaction. So all these chemical reactions are in a pathway. They are in a isomerization. They are in a catabolic pathway. I'm writing over my enzyme here. A catabolic pathway. Alright? They are in a catabolic pathway. It's the step-by-step breakdown of sugar into what will eventually mostly be energy and carbon dioxide. So, again, one chemical reaction just happened here, but the full catabolic pathway is several different reactions with several different enzymatic types of enzymatic reactions. Okay, so what we just saw on the previous slide was a very messy, not great example of a metabolic reaction. So a metabolic reaction or a metabolic pathway, you can use reaction and pathway interchangeably, is what we saw on the previous slide with the breakdown of sucrose. It's also represented here a little more simply. Right, we start with A. On the previous slide, that was sucrose. Use an enzyme to convert A into B, and that B can be one thing, it can be two things. In our previous example, it was glucose and fructose. And we used a particular enzyme to do that, and that enzyme's name was sucrase. And sucrase did hydrolysis. Alright. Now we're going to take that glucose and fructose and convert it into G6P. And the enzyme name that does that, he has a name. We don't have to worry about his name, but there's his name, hexokinase. The reaction type was a phosphorylation reaction, etc. So you get the idea. These pathways, either catabolic or anabolic, happen step by step in little chunks, not all at once. By the way, here's a couple other examples of metabolic pathways. Cell respiration, which is our lecture 9, which we are going to cover next. Photosynthesis is a metabolic pathway. Lactic acid fermentation. Maybe if you work out, you've probably heard of lactic acid and how it builds up in your muscles when you work out. We will talk about that too. Pentose phosphate pathway is another metabolic pathway. Fatty acid beta oxidation pathway is another pathway. Lots of them. Alright. Lots of pathways that we are not going to cover. Just examples. Okay, let's get into our different types of chemical reactions. Here I have the term metabolic reactions. I'm actually not sure I like that term. Now that I think about it, let's replace that term with chemical or enzymatic. Reactions. A little better. Okay? So, your metabolism includes 4 main types of these chemical or enzymatic reactions. One we've already learned all about, hydrolysis and dehydration. We are not going to cover that one again. Save you the agony there. Another one that we're going to learn about is called reduction oxidation reactions, And if you think it sounds familiar, that's because it should. We just looked at reduction and oxidation and chemical potential energy. We're going to come back to that as we promised. We're going to look at exergonic and endergonic reactions. And you actually may have heard these terms already in a slightly different form. Exergonic, you may have heard in chemistry as exothermic, endergonic as endothermic. They mean essentially the same thing. Exergonic simply means the release of energy. Exothermic means the release of heat. But you can put either one on an exam. And we'll learn two types of exergonic and endergonic reactions. And then finally, isomerization reactions. Remember, an isomer is in chemistry something that has the same chemical formula but a different shape. So same number and types of atoms, but a different shape. That's an isomer in chemistry. And an isomerization reaction is simply the conversion of one isomer into another isomer. Alright. Now one thing to keep in mind with these are not completely separate. You can have a reaction that is a reduction reaction that gains energy, that is endergonic. You can have a hydrolysis reaction that is exergonic. You can have an isomerization reaction that is exergonic. Okay, so there's a lot of crossover between these. Okay, so let's look at reduction-oxidation reactions. Very often, reduction-oxidation reactions, because they happen together, a lot of times we just squish the words together, we call them redox reactions. Kind of like Brad and Angelina Jolie become Brangelina. Reduction-oxidation become redox. So remember we said that a chemical reaction is the transfer of energy or electrons, atoms, etc. Reduction and oxidation reactions in particular are a type of chemical reaction that have to do with the transfer of electrons. Reduction in particular means the gain of electrons. Oxidation means the loss of electrons. Now why is reduction called reduction if you're gaining electrons, if you get more stuff, right? Why are you reducing? Remember, electrons have a negative charge. So when you add electrons to an atom, let's take a calcium atom that has a 2 plus charge. Let's add that electron to the calcium atom. It becomes a 1 plus. We are reducing the charge. So when you add electrons, you're reducing something. Could be a single atom like we just showed, or it could be an entire molecule. With oxidation, you are losing electrons. And most of the time, you're losing electrons to this guy, oxygen. Remember, oxygen's very electronegative. He'll take electrons wherever he can get them. So most of the time, when you are losing electrons, you're losing them to oxygen, and that's why this is called oxidation. And here's how I remember this. Down here, oil rig. Oxidation is losing. Reduction is gaining, and we're referring to the electrons here. I've also heard some of my students say, Leo the lion says, Grr, losing electrons oxidizing, gaining is reducing. I don't like that one as much, but if it works for you, go for it. I like oil rig, nice and simple. Okay, so here's the thing about electrons though. They're energy, you can't just get rid of them, you can't destroy them, right? Can't create them from nothing, first law of thermodynamics, right? So that means that if you're losing an electron, if you are losing an electron, someone else has to be gaining it. Your loss is someone else's gain. Right? So reduction and oxidation reactions are always paired. If one molecule is losing, the other is gaining. If one's gaining, someone else somewhere had to lose. kind of like money. So going back to this now, I know you're itching. I know you were going, wait a second, when are we going to come back? Well, here we are, and I realized I forgot oxygen over here. So here's a molecule, and I'm just pulling out the CH bond right there, just for demonstration purposes, okay? This is a fairly reduced molecule, right there, or a fairly reduced bond. You can say a whole molecule is reduced, or just a single bond is reduced. You can say a whole molecule is oxidized, or just a single bond is oxidized. So right here we have a fairly reduced bond, okay? Right here, notice we're bonding with oxygen. Those electrons are spending a little more time around oxygen. That carbon right here has been slightly oxidized. It's had its electrons not fully ripped away, but kinda sorta. And here, ooh, electrons, man. They're really spending some time around oxygen right there. Lots of electrons, 2 pairs of electrons, because we have a double bond. So this oxygen has really been pretty heavily oxidized. This one right here, oh man, carbon's got almost nothing left. Okay, carbon's got almost nothing left, sorry. Keep doing that. This carbon is quite heavily oxidized, and then we get here, carbon has nothing left to live for. Has no electrons. All its electrons are belonging to the oxygens. This carbon right here is fully oxidized. Alright, so when molecules are oxidized, they usually gain an oxygen, or they gain, so here we gained an oxygen, here we just gained another bond to the oxygen. Either one can happen. In contrast, when you're reducing, when you're adding electrons, you may notice that you're adding hydrogens. So you gained a hydrogen. You gained a hydrogen. Here, going from carbon dioxide to the C double bond OH, you gained a hydrogen. You have another hydrogen. So usually when you reduce in biology, you usually gain a hydrogen along with the electrons. And it's because electrons really don't like to come alone. So usually they need a carrier, and that carrier is hydrogen. When you study this in chemistry, if you guys ever get that far in chemistry, you will not look at oxygen and hydrogen, you're just going to look at the movement of electrons. So just keep that in mind when you get to your chemistry class. We're looking at this as it pertains to biology. Now one thing that's kind of interesting, not something you need to know, but a little tidbit. Because oxygen is abundant in the air, lots of food starts spoiling via oxidation. Oxygen bonds to the molecules in your food and starts tugging on its electrons, or even ripping away electrons, as soon as it can get to the food, so as soon as the seal on the container of the food is broken. This is especially what happens with oil. Oil oxidizes very easily, and it gets that kind of stale oil smell and taste. Right? Anything with lots of fats oxidizes very easily. Okay. Now, there's a little bit of difficulty here in these redox reactions in cells. Ideally, what we'd like to do is just rip away electrons from our food molecule. So we'd love to take a juicy glucose. Let me just take one of the bonds in glucose. Say a COH right there. Okay, that's one of the bonds in glucose. We'd really just love to rip away those electrons. But here's the thing about electrons. Think of them like kids, alright? Destructive little kids. The kids that are like in their terrible twos and throwing temper tantrums and like writing on the walls and ripping up your couch and everything. Alright, you can tell I've taken care of kids. So we can't just let those electrons go by themselves in the cell. We can't just rip them away and just let them go free. So we have to have sort of a babysitter for them. So we call those babysitters electron carriers. And these electron carriers are temporary, right? You pay the babysitter and then, he or she goes home at the end of the night. Babysitter doesn't have to worry about the kid after a while. They're just temporary. But they do their job. They babysit the electrons until we can ship those electrons elsewhere. So we have two electron babysitters, NADH and FADH2. Heck of a name, huh? So both these are big complex molecules that act as these electron carriers, and they're temporary storage molecules for electrons. So what we will do is we will take in glucose or fat, any molecule really, rip away some of the electrons through an oxidation reaction. So we're ripping away electrons. That means we are oxidizing because reduction is gaining. And we've got to give those electrons to something as a temporary storage. We give them to either NADH or FADH2. Now this reaction is different from hydrolysis. It's different from condensation. Hydrolysis and condensation were just breaking bonds. Here we are ripping away electrons. And we'll see what these look like on the next slide. So NADH, the way it's written out right here, notice it has the H. And remember when we said that when we have an H attached, that means we've added electrons. So NADH has electrons. We see that as the reduced form. When we take electrons away from NADH, when the babysitter goes home for the night, we are oxidizing NADH. We're removing its electrons, and it becomes NAD+. So think about it this way. Think of a number line. Right in the middle is 0. Okay. Here's +1. Here's, oops, I was going to say +2, but that is not +2. There's -1. NADH, in its reduced form, is right there. When you take an electron away, you're taking away a negative charge. It's going to become plus. Okay? So that's why it becomes NAD+. So we say that is the oxidized form. And that NAD+ can be reused. You can call that babysitter again for another night. You can reduce it again back to NADH. So this is a reusable electron carrier. We have these floating in our cells by the millions. FAD and FADH2 work the same way. Notice FAD doesn't have that plus. We won't get into why. It's an organic chemistry thing. So FAD is the oxidized form. FADH2 is the reduced form. Again, we're adding hydrogens, so we are adding electrons. So now let's look at FAD and NAD+. Quick quiz, are FAD and NAD+ the oxidized forms or the reduced forms? Remember, they are the oxidized forms. They're the oxidized forms. Going back. Alright, they're the oxidized forms. Okay, so here's what these molecules actually look like. And keep in mind you got millions of these floating around in your cells all the time. Look at the names. FAD first. Flavin, adenine, dinucleotide. Ooh, okay. We may recognize some of this. Remember when we talk about the structure of a nucleotide, there's 3 parts. There's the 5-carbon sugar. There's the phosphate group. And I'm running out of room here. And then the 3rd part is the nitrogenous base. The nitrogenous base. Alright. So when we look at FAD in particular, here's one form of FAD. This is the oxidized form. This one over here is the reduced form. Okay. Here we have ribose, our 5-carbon sugar. There's adenine, there's nitrogenous base, and there's our phosphate group. But look, there's another one, another phosphate group. This is a sugar right here, I promise you. And then this is a nitrogenous base. There's 2 nucleotides, hence dinucleotide. Adenine is one of the nitrogenous bases. Flavin is another nitrogenous base that we didn't learn about in the beginning of the quarter. So that's why it's called FAD. It has this weird, big, complex structure. When we reduce it, when we add electrons, remember we add a hydrogen, we specifically add electrons right there and right there. Okay, so we actually add 2 pairs of electrons to FAD. Here with NAD+, same thing, nicotinamide adenine dinucleotide. Alright, so here's the oxidized form of that nicotinamide adenine dinucleotide. 5 carbon sugar, nitrogenous base, phosphate group. That's 1 nucleotide. Phosphate group, 5 carbon sugar, nitrogenous base. This is the nicotinamide nitrogenous base right there. There it is in its oxidized form. When it's reduced, we add the hydrogen right there. Now you may be wondering where we get these from. Well, FAD is actually derived from riboflavin. And again, if you were one of those nerdy kids who read the back of your cereal box when you were little, right, you noticed that there is a particular vitamin called riboflavin. And I actually do not know if this is the case. I know it's one of our vitamin Bs. I don't know if it's B12 or B6 or B whatever. I just know it's one of them. So you need one of your vitamin Bs so you can generate your electron carriers in your cells. Same thing with niacin. I think that's another vitamin B too. So niacin you need in order to generate NAD and NADH in your cells. What was really interesting, actually, I had a student who, a couple years ago, who had a daughter who had a mitochondrial disorder. So her daughter's mitochondria didn't quite work properly. And as we know, the mitochondria are the powerhouse of the cell. They are where the breakdown of sugars happens. And since we're breaking down sugars, we're doing redox reactions. And since we're doing redox reactions, we need these electron carriers. Well, in the case of her daughter, because her mitochondria didn't quite work properly, her daughter had to have a huge intake of niacin and riboflavin in order to kind of make up for the mitochondria not quite getting all the way there. Okay, so here's what happens then. We take a molecule like this, and this molecule right here, remember, it's the organic chemistry lazy person shorthand version of the molecule. Keep in mind, there's a CH3 right here. There's a CH3 right there. There's a carbon right in the middle. We're going to oxidize this carbon right there. We're going to attach an OH and remove the CH3. So we're oxidizing this carbon. We're stripping electrons away. We're going to transfer those electrons to an electron carrier. We won't worry about which one we're using, at least not quite yet. It's going to be one of these two, either NAD+ or FAD. And if it's NAD+, that NAD+ is going to be reduced to NADH. If it's FAD, that FAD is going to be reduced to FADH2. So keep in mind here, I'm following my oxidation ladder. All right, if I go all the way back even further. If I'm following that oxidation ladder, I'm going from this stage right here to this stage right here. I'm going from that C double bond O to C double bond O OH. Okay. I'm going from C double bond O to C double bond O OH. I'm oxidizing, therefore I have to be reducing, and I'm going to be reducing my electron carriers. Alright, so I'm actually going to upload this as its own separate video because I think, yeah, we're hitting the 50-minute mark, and my guess is you are probably ready to go take a break and or go get a really strong drink. Okay? So I will meet you back here when you're ready for part 2.